process of fertilization. Fusion of the male or sperm and female or ovum gametes to form a zygote is called fertilization. In lower vertebrates, fish and amphibians, fertilization normally occurs outside the body of the female, generally in water, and is called external fertilization. But in higher vertebrates, reptiles and mammals, fertilization occurs within the body of the female and is called internal fertilization. Fertilization brings together maternal and paternal chromosomes and thus restores the diploid number of chromosomes. The process is of genetic significance as by bringing together the chromosomes from two different parents, variability is introduced. It is also of physiological importance as egg is activated by sperm to initiate its further development. The whole process of fertilization in humans or mammals can be divided into 5 stages. 1. Maturation and capacitation of spermatozoa. 4. Passage of spermatozoa in female genital tract. And 5. Ovum-spermatozoa interaction in the fallopian tube. The spermatozoa pass through the male genital passage and undergo maturation, but acquire motility only after reaching the epididymis. Secretions of the epididymis, seminal vesicles and prostrate have a stimulating effect on spermatozoa, but they become fully motile only after ejaculation. Spermatozoa acquire the ability to fertilize the ovum only after they have been in the female genital tract for some time. This final step in their maturation is known as capacitation. The ovum in humans is alecythal, that is, almost devoid of yolk but with a bulky cytoplasm and a centrally located nucleus. When shed from the ovary, it is not fully mature, but a secondary oocyte which remains surrounded by a secreted, transparent, non-cellular layer called zona pellucida, that is, elongated follicle cells are present outside this layer. Some of these cells remain stuck to the outside of the zona pellucida. Between the cell membrane and zona pellucida is a distinct space called perivitelline space, where lies the first polar body which separates from the ovum at the first meiotic division. The region of the ovum which extrudes polar bodies and receives sperm is called animal pole. The pole of the ovum opposite to the animal pole is called vegetal pole. The passageway for semen to the external world is urethra, a channel inside the penis that transfers spermatozoa from a male to the female. During coitus, the parasympathetic nerves of the penis are stimulated. That causes tremendous vasodilation. At the same time, the venous drainage is also obstructed. Arterial dilation in conjunction with venous obstruction leads to the retention of blood within the penis, which in turn causes it to enlarge and harden, a phenomenon called erection. Erection facilitates the limp penis to penetrate the female vagina. The climax of the act is orgasm, during which heart rate increases, blood pressure rises, and emotional excitement reaches a climax. Orgasm begins with emission, movement of spermatozoa into the urethra by rhythmic contractions of muscles in the testis, epididymis, and vasa deferentia. Simultaneously, the muscles of prostrate gland and seminal vesicles contract, pouring their secretions into the urethra. When the penis is filled with spermatozoa and secretions, contraction of muscles at its base causes ejaculation. After ejaculation, the erection of the penis subsides. Enormous number of spermatozoa, about 400 million, are contained in an average ejaculation. During coitus, semen containing millions of spermatozoa is deposited in the vagina. Since the site of fertilization is the fallopian tube, spermatozoa have to travel a great distance through the female genital tract beset with chemical hazards. Thus, after entering the vagina, spermatozoa move into the female genital tract and reach the uterine tubes. Most spermatozoa cannot survive for long in the vagina due to its normal acid reaction. The surviving ones migrate upwards and enter the uterus through the cervix. Creeping along the endometrial surface, the remaining spermatozoa try to reach the fallopian tubes. In favorable phase of menstrual cycle, they can survive for about 72 hours in the uterus and fallopian tubes. Only one spermatozoon out of millions reaches the ovum in the fallopian tube by overcoming several mechanical obstacles in its course of path. The ovum at the secondary oocyte stage extrudes from the ovary and trickles down the fallopian tube, being encircled by follicle cells, corona radiata. The follicle cells are glued together by a mucopolysaccharide called hyaluronic acid. Fertilization of the ovum occurs in the ampulla of the fallopian tube. On reaching the fallopian tube, the spermatozoon undergoes some changes that are collectively known as sperm capacitation, which has important features. 1. Activated motility, by which the gentle movement of the spermatozoon's tail changes into a whip-like movement. And 2. The acrosomal membrane is lost so that the enzymes at its head region are exposed. The spermatozoon then releases an enzyme, hyaluronidase, sperm lysin, that helps to remove follicle cells from the ovum and facilitates the entry of the spermatozoon into the ovum. After the entry of the spermatozoon into the ovum, its head becomes the male pronucleus, whereas the chromatin of the ovum forms the female pronucleus. Eventually, both fuse together and a zygote is thus formed. In the fertilized ovum, the usual number of 46 chromosomes in a human cell is restored through equal contribution from the spermatozoon and the ovum. Within hours of conception, the zygote undergoes cell division. The presence of progesterone prevents further female eggs being produced. Within the first week after conception, the fertilized egg travels towards the uterus, where the continued growth of the zygote will occur in the form of an embryo. Therefore, the process of fertilization is not just a wonder in the world of science, but it is also the only answer to the very existence of humans on the face of this planet.